Hi, I'm Sarah Keller, and I'm delighted to tell you about my group's research. We are experimentalists who investigate a wide range of biophysical questions that involve self-assembly, complex fluids, and soft matter. Most students who are entering graduate school have never done research in these areas. I hadn't. But you already have a ton of intuition about these topics from your experience with salad dressing. The oil and vinegar in salad dressing are two coexisting liquid phases. If you shake salad dressing, you get a bunch of little spheres that diffuse over time with Brownian motion until they collide and coalesce. The experimental systems that we investigate in our laboratory tend to be cell membranes, or artificial versions of those membranes. You can think of the small spheres that you see here on the membrane in the same way that you thought of the small spheres in the salad dressing. This system is amazingly rich. Think of all the questions you can ask. How do the domains diffuse? How fast do they coalesce? What parameters like viscosity and edge energy set these timescales? Do both sides of the membrane behave in the same way? What if the membrane is next to a surface? What happens when the membrane is near a miscibility critical point? Do membranes in living cells do the same thing? If so, what effect do membrane domains have on the way that a cell functions? The list is huge. Our lab is driven by questions like these rather than focused on a particular technique. That means that we're going to learn whatever technique we need in order to answer our questions. We usually start with fluorescence microscopy, which we do in our own lab. If we need equipment that isn't in our lab, we use one of UW's many user facilities, or we collaborate at UW and also with scientists at other institutions. The University of Washington is a fantastic place to collaborate because it is so interdisciplinary and collegial. I especially appreciate collaborating with theorists because we don't do theory or computation. At the bottom of this slide, I've listed some of the techniques that lab members have used. I don't know exactly what technique you would use if you joined the lab. It depends on the types of questions you're most interested in answering. You'll notice that the way that I explained our research did not assume that you know any biology. Graduate students in our lab came with undergraduate research experience in a wide range of fields: astrophysics, biochemistry, material science, physical chemistry, mathematics, inorganic chemistry, high-energy physics, and also biophysics. Over time, about half of the PhD students in our lab have earned their degrees through UW's chemistry program and half in the physics program. I'm listed as faculty in both departments. I look forward to meeting you and telling you more about the research we're doing now. Most importantly, I hope you'll talk with the students in the group, some of whom are shown here, not just because they are great, but because they'll be honest with you. I especially encourage you to talk with them when I'm not around so that they feel free to tell you what they like best about the group and also what they like least about the group. When a team works together, it makes the lab more productive and more fun. Your job is to find a team where you will get the best work done and be the most happy. I hope that place turns out to be at the University of Washington, and I'm looking forward to talking with you.